It's my pleasure today to tell you about the research related to phage therapy as alternative approach to fight antibiotic resistance, resistance microbial pathogen, as we started recently here at MIPT. And I hope that my information that I give you in my talk will be addition to what we just hear from the previous speaker talk. By World Health Organization estimates, the antibiotic resistance today is a global crisis for the health and wealth of nations. And among the three bacteria, Klebsiella, E. coli, Staphylococcus, they were selected from the large group of the 7 that WHO highlighted as being as a key antibiotic resistance concerns. And all 3 of them, they are on the matter of threats very close to the border public health issues such as HIV, tuberculosis, and malaria. And however, over the last 30 years, no major new types of antibiotics have been developed. The AMR death contributed to the 2050 will be reached to 10 million of people. And there is, as you see, as the biggest among the other threats of human health. And also AMR death distributed geographically. As you see, the major in Asia and Africa reach more than 4 million people. Increase in infection rates alone will reach to the 150 million people dying, presumably, and also reduce the GDP up to 15 trillion US dollars from the time now to the 2050. And interesting to know that particular countries at risk include India, Nigeria, Indonesia, related to the malaria infections, and Russia associated with a high risk of tuberculosis. At the same time, continue a race in resistance by 2050 would lead to 10 million people dying, as I said, and also will bring to, actually will impact on world GDP in trillions of dollars. And taking this, all the problems and the considerations, WHO started in 2014 the leadership in global action plan to tackle AMR between 194 countries globally. And by Center of Disease Control, the 17 bacterial pathogens were taken under consideration, and they have been divided into 3 major groups of urgent threats, serious threats, and concerning threats. And also, however, the new antibiotic developments has commercial problems, which are associated with the fact that development of the new antibiotics markedly declined over the past several years, decades even, and we have to deal with a slight modification of the existing drugs rather than new classes of antibiotics. And also the large pharmaceutical companies have abandoned antibiotic development in the favor of medicine with a great potential returns in investment. overall, the current pipelines of the antibiotics development is inadequate to the combat antibiotic resistance, and will request more than 800 millions to result in over the age of the new approved antibiotics per year. Taking what was mentioned above, is a health threat by AMR a return to the dark age of medicine? No, because we believe that phage therapy can be used as an alternative approach to combat infection caused by microbial pathogens, because the phages are ancient, ubiquitous, and we've been constantly surrounding them. We're born with the bacteriophages, and bacteriophages has several of positive advantages compared with antibiotics use. The first phage therapy started in 1915. In 1915, then Felix Darrell first time used the bacteriophage preparations to treat avian typhosis outbreak in France, and had a very first positive result when all chicken have been survived. In the follow-up years, we have a very very good and positive documented cases of the positive and successful use of the bacteriophages. But as you see from the list, they are all against the animals. However, the first bacteriophages used for human treatment have been done in Russia in 1930s for treat Russian troops in the Soviet Union against the dysentery, gangrene, and severe wounds during the war. And also another, later on, almost 70 years later, was very good, very successful result of the treatment with bacteriophages. The victim who has for whom nothing have been helped during the 3 weeks of treatment until they start to use the bacteriophages. And this surgery and treatment with bacteriophages have been done in Eliava Bacteriophage Institute in Georgia, former Russia. And another also impressive result, the successful recovery after 2 months of treatment. Some, as you see, very severe mine exposed injury. Bacteriophages, as I said, have a lot of number of advantages in preparations. And at present time, here is we are glad to say that many companies around the world already produce the preparations based on bacteriophages, and they are in Europe, Israel, Australia, USA. And then on top of my this table, you see the very successful Russian company Microgene, which produced 17. We have on the market 17 preparations used for human, and also the second for them is pharmaceuticals company in Georgia, former Russia, that also successfully produced many different bacteriophages preparations. But I have to mention that most of them, they have a prophylactics intention, and use most of them, but not all, but used topically with topical application. what Russia has on the market now, that it's important to say that Russia has officially permitted phage therapy. In bacteriophages preparations used for the treatment and prevention and treatment acute intestinal infections, and also used for treatment prophylactic septic hospital diseases such as very severe caused by Pseudomonas, Staphylococcus, Streptococcus, Escherichia, Proteus, and Enterobacter. And in 2014, the sale of bacteriophage preparations in Russia will increase by 9% compared with the previous year. And in the end of 1960, planned to launch several major new production plants of bacteriophage in Russia. And nevertheless, still we need more bacteriophage preparations to develop and use for health, human health treatment, and first of all to be recognized as a prescribed medicine instead of using only as a prophylactics. Taking all of this problem, we have the phage therapy now under consideration. We proposed the in vitro study of phage therapy, and we develop here at MIT cell model phage therapy as an alternative approach for preclinical trial procedure. And we hope that development of this package of recommendation will address to bacteriophages-based drug developers in order to assess the efficacy of new therapeutical preparations to combat antibiotic resistance of microbial pathogens. Okay, this has to be this one. No. We were able to do this proposal based on our platform, have acceptable in our lab. This is innovative equipment, Image Express macro Excel system. It's produced by Molecular Devices USA company, and this will be our technological platform for high phenotypical screening of our cells model system development. What we will do, and we will propose to in this step in vitro cell model development, after development of this system, we will have evidence of phage ability to decline pathogenic microbial agent population in infected human cells. We will be able to get the wide range of dose testing drugs in order to determine the best therapeutic dose. And we will be able to get the full set of bacteriophage cocktail toxicity assessment and best arrangement of pharmacokinetics parameters. Such tests will assist pharmaceutical company to decide them whether or not the drug candidates have scientific merit to further development, and also will assist the drug developers with the confident of ability taking decision, go or not to go, for clinical trials phases based on the relevant human models instead of relying on sometimes inconsistent animal preclinical data result. What we will do high content screening, infect the human cells by medically important species, microorganism Klebsiella, Pseudomonas, Acinetobacter, and Streptococcus as a major threat in antibiotic resistance, and then treated the biophage preparations. The next step will be screen phage treated cell culture on the biological properties improvement, including production of cell inflammation, apoptotic factors, cell viability, morphological characteristics, and other biological markers. We will obtain quantitative data of phage therapy efficacy based on statistical data analysis of cell model. And then, finally, we develop bioanalytical methods for pharmacodynamics and pharmacokinetics of the phage-based drug assessment. Okay, so what is this? And we think that this is a cell model system. For our knowledge, this in vitro cell model system is never used before in different researchers, by different researchers, and they're pretty much innovative. And based on the result we will get, we can propose experimental cell model as a base technology platform for future preclinical phage therapy test system development, and also significantly improved evidence gathered by our data analysis. They will serve as a proof for recognition of phage preparations as a pharmaceutical drugs for official use in medical practice. This research, we just started in collaboration with several Russian organizations, such as Obolensk Institute of Gabrichevsky Institute. It's a Institute of Microbiology, and also Institute Bioorganic Chemistry. So, and the most of the people will do work for this as our laboratory of innovative drug development, students and scientists. Thank you. This is.